So we have finally gotten into that part of biology that you thought you were getting into when you signed up for the course, a semester and a half ago. We're talking about animals. We finally got into, you understand the chemistry, you understand the evolutionary background, you understand basic cellular biology and how these cells hold on to each other. We sort of began to allude to animals when we talked about choanocytes, the choanoflagellates with protists. Now we're getting into that next level of multicellular animals. So, characteristics of animals. Guys, and in today's series of lectures, I'm going to go through a whole bunch of stuff. Not everything you have to write down. Don't try to keep up because there's going to be a ton. I will tell you specifically, this is important. For that, this is important. Generally, whenever you see me put a list up on the board, think, I'm going to have to know that. Characteristics of animals. All animals are multicellular. They're all eukaryotic, heterotrophic. They have no cell walls. They all have nervous tissue. They can move and reproduce sexually. They have an extracellular matrix, Hox genes, and similar ribosomal RNA. Now, you're probably thinking, Chris, stop. I haven't written all that down. Don't worry. I have a whole bunch of slides with all of these at the top of the slide, and we're going to go through each one. So for the first one, all animals are multicellular. Even the simplest of animal is made up of many, many distinct cells. This is Daphnia magna. Daphnia are these great little water fleas. They live in literally a droplet of water. Very simple. You can see through them. You can see all their eggs. They are made up of cells. They are multicellular. They're all heterotrophs. What does it mean to be heterotrophic? Do you guys remember? Right, you're going to take in organic molecules from the environment that other plants might make, or from an animal. You have to ingest new materials. So all animals are heterotrophic. They have no cell walls. What kind of organisms have cell walls? We talked about, 3 types of organisms. Fungi, bacteria, and plants, yeah. Not so with animals. Animals may seem like they got a cell wall sometimes because they got these hard chitinous exoskeletons like this lobster up here. Usually if an animal has a hard carapace or claws, it's made up of keratin, which is almost like really, really tightly woven hairs. They have nervous tissue, so they have a nervous system. That's what we're going to get into next time, the different systems. This allows animals to react to stimuli. They have cells that are specialized to react to their environment, to send signals from one place to another very, very quickly. All animals can move. Most animals have muscles. Not all. Most animals have muscles. And those muscles are typically attached to some sort of support system that allow them to move at one stage of their life or another. Now, some animals will stay in one spot for the entirety of their lives, but not all. They all reproduce sexually. Now, there's some animals that can reproduce asexually as well. I don't know, if you take a flatworm and cut it in half, boom, you got 2 flatworms. But all animals are sexually reproductive at some point in their lives. In an egg, binds to a sperm, and they create a diploid zygote. They have what's known as an extracellular matrix. The inside of the cell is interesting. You learned a lot about the inside of the cell last semester. What you didn't talk so much about is what's happening between the cells. In an animal, you have this area between cells called an extracellular matrix where cells can interact with each other. They all have Hox genes, H-O-X genes. Hox genes are really cool, and I'm a geneticist, so I think they're super, super cool. These are genes that are responsible for body plans, that make you have a left side and a right side, that indicate this is, arms or antenna should grow. Not you don't grow antenna, but you got some genes in there that might have originally been about for antenna. They give you your body axis patterns. They tell you when to grow this limb versus that limb. The body plan genes. They all have similar ribosomal RNA. I put a picture of a cuttlefish on here, or a squid, because I used to, when we talked about bacteria, I had a picture of ribosomal structures, and you guys didn't really like that much. So it's a cute squid. But those ribosomal subunits among animals are super highly conserved to build proteins. So ribosomes are very, very similar among animals. Now, true multicellular animal life began in what's known as the Cambrian era. It happened during what's called the Cambrian explosion. Basically, at one point in history, 590 million years ago, we had no real evidence of multicellular life. We had a lot of stromatolites that were built from bacteria, but nothing else really that we saw. And then suddenly, within a very, very short time frame, life was everywhere. There was tons of multicellular life. It was basically like somebody flicked on the light switch and life was there. Multicellular life. Again, we know there was protists and early eukaryotes, some bacteria through the seas, but then quite suddenly, about 590 million years ago, we had animal life. There are a few reasons this may have happened. One is that during this time frame, there was extremely high ultraviolet radiation. Ultraviolet radiation, as you guys know, causes mutations. It causes changes in different organisms, in their DNA. It's also expected at this point that the Hox gene developed. Now again, Hox genes are about body plans. So if the Hox gene develops and there's this rapid mutation, you get lots and lots and lots of different forms coming about. Evolution can act on different alleles. So you have plentiful alleles, new variation, and it led to very, very, very quick evolutionary jumps. This was known as the Red Queen theory. There was a Lewis Carroll's Alice in Wonderland, where the Red Queen turned to Alice and said, you have to run as fast as you can just to stay in the same spot. And I don't know about you guys, but that really sort of resonates because I feel like I'm trying as best I can to get ahead in life. I'm running as fast as I possibly can, and I'm probably getting nowhere. Welcome to life. But that was Lewis Carroll, what she was getting across, and sort of summarizes evolutionary theory. Evolution is a net zero gain. For every step forward one type of organism makes, another organism will take another step. Every time a predator develops some sort of new attack method, a prey develops a new defense. And they go back and forth. It's an evolutionary arms race that never ends. So far, so good? So these are all three reasons why life may have happened during that Cambrian explosion. High ultraviolet radiation, Hox gene development, and the very rapid evolutionary competition between organisms. The first animals were thought to be, related to the quantiflagellate. That we talked about back when we talked about protists. The quantiflagellate had two super important genes that gave rise to multicellularity. What were the two genes required for multicellularity? Yes?
Extracellular communication and the one that helps the cells stick to each other.
Yes, so extracellular communication. Cells have to talk to each other, and cellular adhesion. Cells have to stick to each other. That quantiflagellate had those, and it ended up giving rise to the earliest of animals. We can see the structure of the quantiflagellate up here, reflected in, see over here, the quanocyte cell of the sponge. They are exactly the same. Genetically, they're the same. This is not convergent evolution. This is direct evolution. A unicellular organism becoming multicellular. Questions so far? So what we're going to do basically for the rest of these next several lectures is classify these animals. Animals are primarily classified by their body plans. They've got tissues, they've got symmetry of some sort. We might look at their embryonic development. I left these two at the end, segmentation and the presence of a coelom, because probably about 5-10 years ago this was thought to be very important. It's not anymore. It's basically, this is an example of convergent evolution in animals. So really we're going to focus on tissue, symmetry, and embryonic development. Tissues, as we discussed with plants, are cells of a specialized type working together. So it's one cell type working with other cells of the same type. Tissues come together to form organs. So big classification is, do you have tissue or do you not have tissue? That's like the first big split among animals. So we have parazoans and eumetazoans. Parazoans, like sponges, do not have tissues. They don't have specialized groups of cells working together. They have individual cells working together. Eumetazoans, on the other hand, are like you, and apparently like this hydra. They have specialized tissues and organs. So once you've separated them between yes tissue, no tissue, parazoan, eumetazoan, you take the eumetazoans and you start splitting them up by symmetry. So are they bilaterally symmetrical or radially symmetrical? So radiata display radial symmetry. You can cut them along many different axes, and both sides will look the same. Heck, you can cut them into individual chunks, and both sides look the same. Bilateral symmetry is like you and this fly. You can only cut it in one direction. You cut it in half. You got a left side and a right side, and they look similar. They're mirror images of each other. If you cut it down the middle, in the other direction, they won't look the same. So there's only two bilateral, two sides. So we split them up. We look at radiata. That symmetry is formed during embryonic development. All right, so here's the way embryos form. You have a fertilized egg. So an egg is fertilized by a sperm. You get an N plus an N, you get 2N. Talked about that during meiosis and all that in Bio 101. From there, the zygote begins to undergo many rounds of mitosis. It splits, creates identical cells. So you go from a zygote to undergo, round after round of cell division, and then you get what's known as a, so from a zygote to a blastula, and then from a blastula, which is a hollow ball of cells, to a gastrula, which is a hollow ball of cells that has that's been invaginated, has a hole at the bottom. So you get your zygote. The zygote undergoes many rounds of mitosis to produce a big hollow ball. That big hollow sphere is known as a blastula. So far, so good? The blastula then sort of folds in on itself. Imagine, if you will, a balloon, and you take your fist and you push it up through the balloon, so you've got this sort of outside and then an inside cavity. That's gastrulation. And this forms the beginning of, in some animals, of a simple mouth. Think of a, what are those called? Jellyfish. So far, so good? Blastula leads to gastrula. The gastrula then develops multiple layers. You've got an outside layer, and an inside layer, and then this little middle layer right here. So the outside is called the ectoderm. Ecto is outside. Then you got the mesoderm in the middle. That's going to give rise to a bunch of muscles and things. And then finally you have the endoderm, the inside. That's sort of the lining of the digestive system. So, endoderm, digestive system. Your body, as we will discover when we dissect a pig, is a series of tubes. And those tubes pump all sorts of fun liquids through your body. The main tube goes from your mouth all the way down to your stomach, and then does all these twists and turns in your small intestine, and then loops back up through your large intestine and right out through your anus. You have this big tube. You could literally put something from one end of that tube all the way to the other end of the tube if you had a long enough, basically a long enough camera, because you're going to stick a camera down your throat or something. Doctors do it. The mesoderm forms the muscles, the tissues, and the organs, anything that's not part of your digestive system. And then the ectoderm is going to be that outer covering, the epidermis, as well as your nervous tissue. Endoderm, mesoderm, ectoderm. Questions? Say that again, please.
What type of pig are we dissecting?
What type of pig? A baby one.
But like,
I have no idea what type of pig. Sorry. I'm not big on pig delineations. Whichever kind of pig it is that makes you not upset that we're dissecting it, that's the kind.
okay.
So, there's a long time ago, as I mentioned, that we started using the coelom and the segmentation as a way of classifying animals. This whole idea is it's a historical classification method. You don't need to know it. I think the book still might talk about it, but all the recent literature says it doesn't work. So I'm not going to make you learn it. So now we've got how we classify animals. One very simple type of animal is radiata. Radiata are a classification of animals that have two cell layers, and only two cell layers. They have an endoderm and they have an ectoderm. There's no mesoderm, which means they don't have muscles, no organs. So there we are. Questions, concerns. So animals have all those common characteristics that we discussed. They're multicellular, and they develop from a zygote to a blastula to a gastrula into different cell layers. Some animals only have 2 layers. Most of them have 3.